Welcome back to the Computational Genomics Lab. Today we are going to continue our journey with genomic ranges, or G-ranges, and look at some of the annotation features of the human genome. This is probably one of the most useful parts of G-ranges, is keeping track of all those layers of starts and stops of all these different features of our genome. Alright, so let's go take a look. We are in 08 still, in lessons. 08 ATACseq pipeline, 05 our genome in G-ranges. Okay, so let's do that, and what we're going to do is read in an annotation file. So you need an annotation, and this is usually human done or human governed on some level. There's Ensemble, there's GenCode. We're going to be using GenCode, which is also connected to Ensemble gene IDs, and there's some connectivity there. It's a bit confusing. Again, it was built and then tried to be connected. But anyways, there's a bunch of genome information, and the GTF file is the annotation file. There's more information all about this. And so let's dig in here. I'm going to clean my environment from our last class. And let's go to 05, our genome in genome ranges. Okay, so I'm going to load my library genomic ranges. If you don't have tidyverse loaded, you might also want to load that as well. Again, there's more detail written down here about the importance of annotation files. And here on line 49, we're going to load in M25, which corresponds to mouse MM10. The genome is version 10. The annotation is version 25. That's what that difference is there, and that's in the genome folder up at the main directory of the class here. Here under master class we have this genomes folder. Okay, go back to working directory. Okay, so let's import this file. Oh, it's going to take a minute, by the way. That's right. So I'm going to pause the video here. This is going to take about 2 minutes. See you then. Excellent. It has now loaded, and it did take a couple of minutes. But now, in this object in our environment, GenCode GR, where GR stands for G-ranges, we have this huge meta list of information about the mouse genome in this case. And this @element data has the most things like that we're going to want, like gene type, gene name, gene ID, and those kinds of things. So let's make a handy table so that we can have it moving forward of annotations we might want to keep for all the genes. And it's a nice little table to have of all the features of genes, like how many exons, how big it is, etc. We'll go through all that. Okay, so let's start that by just taking a peek at the seq names of GenCode. And that just tells you the levels. The seq names is the chromosome slot that it's keeping track of. And we can table that and see the length of the chromosomes as well. We could do table things. I'll let you play with these. There's no real important concept here. You don't really, the seq names, just to check that you have the 22nd index of the GenCode GR seq names is the mitochondrial chromosome here. Not too interesting yet, but just to show you, this is where it starts to get interesting. So now if we go GenCode, if we open this object here, it's hard to see these things, but if we look at @element data and then @list data, we see all these things we're going to want, like transcript ID, etc., etc. So that's where we want to get to, and so that's what we're doing here. Is we're saying @element data in the gene ID column. So we didn't go @list data. It's complicated, but this is the way we index into the thing we want. But we can see what those things are in this list data, like we just did gene ID here. And so we might want gene type and other things like that. So let's look at the first 100 elements of gene name. And here's the ID names. They look a little different than what we saw in our RNA-seq data. It's okay, though. We have conversion tables. These are from Riken genome annotations. Okay, so then we can also look at transcript name column. And we see it's like Riken. And the the numbers here are the same, so O5C1 here and O5C1 here, and they're the same, but there's a dash. That means they have a different transcript isoform. So it means that, like, instead of maybe just having 2 exons and the other transcript ID, there's a 3rd exon that's spliced in. Okay, so you need to think about how you want to do your analyses, if you want to do them on a transcript level, gene level, etc. And that takes some intuition and some advice. I would just go by transcripts on a gene name level at first, and then you can dig in deeper into the isoform parts as you advance. Okay, so transcript name. If we look at this at row names, not interesting. So it's really all in this at element data is where we're indexing, and then we're using the cache sign to grab things. And here we're doing a tidy verse, doing a table, just a base R command of this indexed column gene name, and we're making it as a data frame here using our tidy pipe to push it over there, and we're going to call it GenCode info. Okay, there it is. We have 55,000 things in there. Yep, and 2 observations, so we can look at that. And since it's a data frame, we got these things. We have the gene name and something that is not as great of an idea. So let's make a more clean table. That was just to show you can pull these things out to make a data frame. Okay, so we're going to call this thing called GenCode GR length. It's kind of arbitrary name. It's just going to have some features we want to keep. So we just have to do this indexing once, and then we have all these features for the rest of our analyses. And that's going to be gene ID, gene name, gene type, type, start, width, and chromosome, just to give you an idea. And we use this data frame function where the column names are in these quotes here, equals, and then the index will fill that column in with information of the gene ID, the gene name, etc. All right, so let's run that. And GenCode, oh, nope, we want this one, GenCode GR length. Okay, so here we have gene ID, a little different ensemble version of the gene name. We have here its actual name, gene type. So here we can see there's protein coding, antisense. There's different types of genes. So there's not just protein coding genes, there's other varieties of genes. And this column tells us here, gene type, what type is it? And you might want to say, I want to look at long non-coding RNAs because they're my favorite. I think they're called link RNA here. And so here they are in this annotation. I can grab just those by indexing a grep or a matching function to that. Where the exons start, and then starts and widths. And only start and width and not end, because if you subtract width from start, you get the end, or add the width to the start, you get the end. All right, okay, so that we're moving along here. I'm going to clean up. I think I opened that thing too many times. Okay, so we have, that's a really handy table we'll probably want to save at the end, and here we go. There we are. We're at the end. Feel free to save this, and then you'll have that nice handy table with all these gene features. And when we look at our RNA-seq data again, or the ATACseq moving forward, we can ask, are they next to long non-coding RNAs or protein-coding genes? Is there a difference? And we can add that level of analysis of any of these genome features. We can add that multiplicity of analyses to our research plan on looking at how docs affects ATACseq data. All right, we'll see you next time. We're going to continue looking at more genome features in a little bit. See you there.